Hello everyone and welcome to the new session of the biology lecture series, and today we'll be discussing the NCERT Exemplar problems for the chapter Molecular Basis of Inheritance. So this is a chapter that is included in the biology NCERT book. Okay, so in the previous session we solved the multiple choice questions, and in this session we'll be solving the very short answer type questions. All right. I would like to mention that all the questions here are completely NCERT based. So let's start with the first question. Okay, the first question is, what is the function of histones in DNA packaging? Okay, so you know about the histones, how many histones are present, and what is the nature of the histones. Okay, so before answering this question, let's understand about the nucleosome. What is nucleosome? You must have heard about this term, nucleosome. Okay, so nucleosome, it is the structural and functional unit of chromatin. Okay, what is nucleosome? It is the structural and functional unit of chromatin. Okay, and what is nucleosome made up of, or what is the component of nucleosome? It is made up of a histone octamer. Means 8 histones are present. Octamer means 8. So means 8 histones are present here in a nucleosome. What are these histones? Okay. So there are pairs of these 4 histones. Okay. So actually 4 histones are present in a nucleosome. H2A, H2B, then we have the H3 and H4. These are the 4 histones and a pair of these. So you can multiply it by 2. Okay, so that's how 8 histones are present and it forms a histone octamer. Okay, and around these histones, DNA is coiled. Okay, all right. So first part was histone octamer. Second part is the DNA which is coiled on these histones. So how does nucleosome look like? Look like this. So we can draw 4 histones. Okay, this one is H2A, this is H2B, this is H3, and this is H4. Okay, and then you can repeat this one again. Okay, and then, okay, and that will form the histone octamer. A pair of these 4 histones are present, and the DNA is coiled around it like this. Okay, so this forms a nucleosome. So nucleosome, you can say it is nothing but DNA coiled around the histone octamer. And what is the length of DNA, or how much DNA is coiled around this? 146 base pair of DNA. 146 base pair of DNA is coiled around this histone octamer. So this is the nucleosome, which is the structure and function unit of the chromatin. Okay. Now, what are the functions of histones in DNA packaging? Okay. So first of all, histones, they help in the DNA packaging by forming nucleosomes. Okay. So first function is histones are the units of octamer that participate in the primary packaging of the DNA. All right. So we have one nucleosome which is attached to another nucleosome. Okay, like this. All right. This is not nucleotide. This is nucleosome. Okay. So they help in the packaging of the DNA. And why packaging of DNA is necessary? All right, because you know in the nucleus there is limited space. So to accommodate this huge amount of DNA in the nucleus, we need to package the DNA. Okay, so these nucleosome will combine. Okay, these nucleosomes They combine to form what? Chromatin. They form chromatin. Okay, this is clear. What is the second function? So histones are basic in nature. Okay, they are positively charged. Histones are positively charged. Okay, and they neutralize the acidic DNA molecules. So DNA is acidic, or we can say it is negatively charged due to the presence of the phosphate group. DNA is negatively charged because of the presence of the phosphate group. So this basic histone protein, they neutralize the acidic DNA molecule, thus stabilizing the DNA or the chromatin. Alright, so these were the functions of the histones. Now let's talk about second question. Now here we have to distinguish between the heterochromatin and the euchromatin, and which one of the two is transcriptionally active. Okay, so let's study first what is chromatin. Chromatin, you have already seen, it is made up of nucleosomes. Nucleosomes are the structure and function unit of chromatin. Okay, so how can we define chromatin? In simple word, chromatin is a mixture of what? DNA and proteins. What kind of proteins? Histone proteins. Okay, and this chromatin. Okay, so this is chromatin. So chromatin is a mixture of DNA and proteins that is found in what? That is found in chromosomes that are present within the nucleus. Okay. All right. Now chromatin, you know, is of two types, heterochromatin and euchromatin. You have to distinguish between them and you have to tell which one is transcriptionally active. Okay. Now let's talk about the answer. Densely packed, okay, and dark chain chromatin regions are known as heterochromatin. Okay, while the loosely packed and light chain regions are known as euchromatin. Alright, this is the first difference between heterochromatin and euchromatin. And which one is transcriptionally active? This was the second part of the question. So euchromatin is transcriptionally active because this is loosely bound, and this is transcribed into mRNA, and then later on mRNA will be translated into proteins. Okay, but if we talk about heterochromatin, so due to very tight coiling, heterochromatin cannot be transcribed and is inert, or it is present in the inactive form. Alright, so this is clear. So transcriptionally active, okay, region is known as the euchromatin. Now coming to the third question. Now the enzyme DNA polymerase in E. coli is a DNA dependent polymerase. Okay, means it required a DNA template, and it also has the ability to proofread the DNA strand being synthesized. Now you have to explain and you have to discuss the dual polymerase activity. Okay, so DNA polymerase. Okay, let's talk about the function or the activity of DNA polymerase. The first activity is the adding of the nucleotide. Means first you can say it has the polymerization activity. It add nucleotides. Okay. Second is it has a proofreading activity. Proofreading activity is due to the 3' to 5' exonuclease activity. Is due to the 3' to 5' exonuclease activity, and adding or polymerization activity is from 5' to 3'. All right. whereas the proofreading or exonuclease activity is from 3' to 5'. So DNA polymerase has both of these activities. You have to explain and you have to discuss these dual properties. Now, DNA polymerase. Okay, in bacteria, there are of three types. So they are of three types. One is DNA polymerase 1. Then we have the DNA polymerase 2. And then we have the DNA polymerase 3. These are the three types of polymerase in bacteria. Okay. Now let's discuss the answer. In bacteria, three types of polymerase are there, and all of them can add nucleotide in 5' to 3' direction. And they also possess the exonuclease activity, which is in 3' to 5' direction. Now, out of the three DNA polymerases, DNA polymerase 3, okay, this plays a major role. Although other polymerase also have this exonucleate activity, but polymerase 3, okay, plays a major role in the proofreading. Okay, polymerase 3 can proofread the newly synthesized strand and senses the wrong base insertion. Okay, so what happens during the exonuclease activity? Or what happens during the proofreading? This DNA polymerase, which has the exonucleate activity, it breaks the phosphodiester bond. You know, the nucleotides are attached with the help of phosphodiester bond. So this breaks the phosphodiester bond between the nucleotide and thus releases the nucleotide, and then it inserts the right nucleotide. Okay, so this is how exonuclease activity takes place, by breaking the phosphodiester bond. And as soon as the phosphodiester bond is broken, the wrong nucleotide is then replaced by the right nucleotide. So this polymerase, it deletes the wrong bases and helps correct the mistake by putting the right nucleotide. All right. Now the only mistake it cannot correct is the substitution of uracil in place of thymine. So this it cannot do. This activity, okay, substitution of uracil in place of thymine, it cannot do. But what can it do? It can repair any damages done to DNA by UV exposure. So UV exposure, they cause mutation. Okay. What they cause? They cause single nucleotide polymorphism. They cause means change in the single nucleotide of the DNA sequence. Okay. So this polymerase 3, they can repair the damage done by the UV exposure. Okay. Or the leftover proofreading mistakes it can also handle. It detects mutation caused by UV, remove the mismatched pairs and put back the right ones. So suppose A pairs with T, and G pairs with C. So instead of suppose, okay, G, here it should be C, but somehow there is a mutation due to UV radiations, okay, and we have the A here. So it will correct this and it will put C instead of A. Okay. Now let's move on to the next question. What is the cause of discontinuous synthesis of DNA on one of the parental strand of the DNA? What happens to these short stretches of synthesized DNA? So here they're talking about the discontinuous synthesis of DNA. Okay. So to answer this question, you should know that the newly synthesized DNA or the polymerization that takes place in the 5' through 3' direction. Okay. So DNA polymerase add nucleotides. They add nucleotides in 5' to 3' direction. All right. Okay. So you must be knowing about the leading strand and the lagging strand. Okay. So let's discuss this first. I'm drawing a rough diagram here. So this is the leading strand. On leading strand, continuous synthesis of DNA takes place. What is the polarity of the leading strand? It is from 3' to 5'. Okay. So here there is a continuous synthesis of the continuous synthesis of DNA from which direction? From 5' to 3' direction. All right, because new nucleotides are added. Okay, new nucleotides are added in 5' to 3' direction, and nucleotides are added where? They are added at 3' OH. Okay, they are added at the 3' OH, and the direction of synthesis is always 5' to 3'. Okay, so this is the continuous synthesis. This is the continuous synthesis. But what happens at the other strand? Okay, this is the other strand. What will be the polarity here? This is 3' to 5'. Okay, now here, this is the lagging strand. Okay, this is known as the lagging strand. This is the lagging strand, and on lagging strand there is discontinuous synthesis of DNA. Okay, so this should be 3'. This is 5'. This is again 3'. This is 5'. So here you can see DNA synthesis is in this direction and it is discontinuous. Okay, so this is known as discontinuous synthesis. This is known as discontinuous synthesis of the DNA. Okay, now these fragments are known as these one. Okay, these are known as the Okazaki fragment. They were discovered by the scientist Okazaki, and they have been named on the discoverer. So these are known as the Okazaki fragment. Here again, an RNA primer is synthesized again and again, and the DNA synthesis is discontinuous. Okay, now this is clear. Now let's look at the answer. The synthesis of DNA always takes place in 5' to 3' direction. In a double-stranded DNA, both strands are antiparallel and they are complementary. What does this mean? I've already showed you one strand was leading strand, other strand was lagging strand, or one is sense strand, other is anti-sense strand. So antiparallel means, so if this is 3' to 5', the other strand will be from 5' to 3'. So both these strands are complementary and they are anti-parallel. Now during DNA synthesis, as both strands act as the template. So both of these strands, they can act as a template. So one will be template strand, other will be non-template strand. Only one strand, that is, which is from 3' to 5', which is we were calling as the leading strand. So this can synthesize the complementary strand in the 5' to 3' direction, as we have seen. Okay, the other strand which is in 5' to 3' direction. Okay, we were calling this as the lagging strand. Okay, we were calling this as the lagging strand. This has to synthesize in small stretches in the opposite direction as the replication fork moves to right. That is why DNA synthesis is discontinuous on one of the parental strands of the DNA. And these stretches these small stretches, they are known as Okazaki fragment. Okay, and they are joined together by DNA ligase enzyme that closes the nick. So the Okazaki fragments are created, these small stretches are created, and later on they are joined together by the enzyme DNA ligase. Okay, and then a complete DNA polynucleotide is made. Now coming to the question number 5. The given below is the sequence of coding strand of DNA in a transcription unit. You already know what is transcription unit. It consists of the promoter, terminator, and the structural genes. So this is the sequence, okay, from 3' to 5'. This is the sequence of the coding strand of the DNA. Now we have to write the sequence of its complementary strand and the sequence of the messenger RNA. Okay, or the okay. So coding sequence, I've already told you the sequence of the coding sequence is similar to the primary transcript, primary mRNA transcript. Okay, now let's have a look at the answer here. So this was the sequence that was given. Now, according to the base complementary rules, you already know A pairs with T and G pairs with C. So keeping this in mind, we will solve this question. So first one is the complementary. So A pairs with T. Okay. A pairs with T, G pairs with, okay. Okay, we have the T, then we have the A, then we have the G, so we will have C. So this is the complementary. This will be the complementary strand. So this was easy. How to make the complementary strand? Okay. Then what will be the sequence of the mRNA? Okay. So this will be this from 5' to 3'. This will be the sequence of the mRNA. Okay. We know the sequence of what? Sequence of the coding strand. Okay. This is equal to the sequence of the RNA. Okay. But in RNA, what is? Thymine is replaced by uracil. Okay. So why we have written 5' to 3'? Okay. It is complementary. So obviously here it will be 5' to 3'. Here also again 5' to 3' because we are making the mRNA transcript. All right. So this will be the sequence here. CGAUU. Okay. So instead of G pairs with C, G pairs with, okay. So this will be the sequence here. All right. Now let's move on to the next question. Here also you can see A. It is similar to the sequence of RNA. Okay. So this sequence will be similar. Okay. So here we will not apply the complementary rule. All right. No complementary base pair rule. Okay. We will not apply complementary base pair rule here because we know that the sequence of the coding strand, okay, is equal to the sequence of the messenger RNA, or you can say sequence of the primary transcript. Okay, but we have to change the polarity here. Okay, this is clear. You can see A and A. Then we have the T, but okay, then instead of T, you will replace it with uracil because in mRNA, thymine is replaced by uracil. Okay, then we have the G. All right, likewise, you can make the sequences. Okay. Now, this RNA uses the base uracil rather than thymine. So in RNA, base pairs are like this. Now, question number 6. What is the DNA polymorphism and what is the importance to study is? So DNA polymorphism is basically it is the presence of two or more variants. It is the presence of two or more variants of a specific DNA sequence. All right. This is known as DNA polymorphism. Okay. Now, one type of DNA polymorphism is single nucleotide polymorphism. Okay, this is known as single nucleotide polymorphism. What does single nucleotide polymorphism means? This means that there is a change in single base pair, or there is a change in the single nucleotide. Okay, there is a single base pair change. Now let's have a look at the answer. The DNA polymorphism refers to the variation in DNA arising through mutation in the non-coding sequences. So in the non-coding sequences mean the sequences that doesn't code for any protein or any product. Okay, these are known as the non-coding sequence, and there is very high rate of mutation. Okay, there is very high rate of variation, you can say, or you can say mutation in these regions because they are not carried forward. Okay, so there is very high rate of mutations, so there is very high rate of variations. There is a lot of variations, and we use this variations to study the to study a lot of things. Okay, we will talk about this. Now, what is the importance here? A special type of polymorphism known as the VNTR, variable number of tandem repeats. Okay, this is composed of repeated copies of the DNA sequence that lie adjacent to one another on the chromosome. So VNTR means variable number of tandem repeats. So these variations, these variations, they lie very close to each other, all right, on the chromosomes. Now, since the polymorphism is the basis of genetic mapping of the human genome, so the mapping of the human genome in the Human Genome Project, this was done on this polymorphism. This was done on the basis of this DNA polymorphism. Therefore, it forms the basis of the DNA fingerprinting also. So this polymorphism, what is the importance? It is the basis of genetic mapping of the human genome, as well as it is the basis of the DNA fingerprinting. So these techniques, these are based on the polymorphism, such as variable number of tandem repeats. Tandem repeats mean they are again they are repeating and they lie adjacent to each other. All right. And there are variable number means there is polymorphism. Okay. Now, single nucleotide polymorphism. If you talk about single nucleotide polymorphism, they are used in locating diseases and tracing of human history as well as in case of paternity testing also. Alright, so this is the importance of single nucleotide polymorphism, and single nucleotide polymorphism can also track the inheritance of diseases associated within the family. They can also trace they can also trace the diseases such as genetic diseases and all. All right. Question number 7. Now, based on our understanding of genetic code, explain the formation of abnormal hemoglobin molecule and what are the known consequences of such a change. All right. So based on understanding of the genetic code, it is a trinucleotide. It is made up of, if we talk about the genetic code, it is made up of, or we can say it is trinucleotide. Means it is made up of, you can say, 3 bases. It is made up of 3 bases. For example, AUG. So it consists of 3 bases. AUG, codes for methionine. So a particular codon. So this is a codon. This codes for a particular amino acid. Okay, and more than one codon can code for one amino acids. That's why we have 64 codons. In total, we have 64 codons, but they code for only 20 amino acids. Okay, this is clear. Now, what happens in here sickle cell anemia? So first, let's take the example of sickle cell anemia. So what happens in sickle cell anemia? So there is a change in the sixth position. So there is a change in the sixth position. There is a mutation in the sixth position of what? Of the beta globulin chain. There is a change in the sixth position of the beta globulin chain, okay, of hemoglobin. Okay, hemoglobin consists of this beta globulin chain. And there is a change in the sixth position of the beta globulin chain of hemoglobin. And what happens at the sixth position? Initially, there is glutamic acid or glutamate. Okay, which is represented by this GAG. This glutamic acid or glutamate is converted into valine due to single nucleotide polymorphism, and valine is represented by GTG. So there is a, you can see, change here. From GAG it becomes GTG. Single base pair change, single nucleotide polymorphism. So because of this, what happens? The shape of the erythrocyte, it becomes sickle shaped, because glutamate is, okay, it is a charged molecule, whereas valine is not charged, it is hydrophobic, and because of this, the erythrocyte, they become sickle shaped. Okay. Now, due to the point mutation, okay, there is a point mutation in the beta globulin chain of hemoglobin molecule. Glutamic acid is replaced by valine at the sixth position, and this results to the sickle shape of the erythrocyte. Because of this erythrocyte, they lose their circular shape and become sickle shape. And because of this, the cells cannot pass through narrow capillaries, and the blood capillaries are clogged, and thus they affect the blood supply to different organs. So this is what happens in sickle cell anemia. Now coming to question number 8. Now sometimes cattle or even human beings, they give birth to their young ones that are having extremely different set of organs, like limbs and position of the eye is different. Okay, so you have to comment what is the reason of that. Why sometimes in cattle or even human beings, okay, that give rise to the young ones. Okay, means they are viviparous. They show vivipary. Vivipary means which give, or which, in which the animals give rise to the young ones. Another one is ovipary, which means where the animals, they lay eggs. Okay. Now you have to comment on this. So what is the reason? So now the formation of these organs and the limbs and the position of the eye, all these are controlled by multiple genes, or they are controlled by set of genes. So what happens? Sometimes there is a mutation in these genes, and due to this mutation, the regulation of this set of genes doesn't take place. There is no regulation. Due to the lack of the regulation, okay, there is no regulation. Due to the lack of the regulation of these set of genes, okay, this results in the development of extremely different set of organs. So let's look at the answer. Sometimes cattle or even human beings, they give birth to their young ones that are having extremely different organs, like limbs and position of the eye, but it happens due to the disturbance in the coordinated regulation of the expression in the set of genes. So I've told you this limb formation and all, it is controlled by the set of genes, and there is a disturbance in the regulation of this set of genes. Okay, so this leads to this abnormal development of the limbs. Now let's move on to the next question. In a nucleus, the number of ribonucleoside triphosphate is 10 times the number of deoxyribonucleoside triphosphate. Okay, but only deoxyribonucleotides are added during the DNA replication. So you have to suggest a mechanism for that. So to answer this question, first you have to understand about the ribonucleoside and the deoxyribonucleoside. So difference is this: deoxy. Here you don't have any deoxy. Here you only have ribonucleoside. But here you have deoxyribonucleoside. Okay. So let's talk about DNA and RNA. So both of them, they are made up of sugar molecule, phosphate group, and nitrogenous bases. So let's talk about the pentose sugar here. In DNA, you have the deoxyribose. Whereas RNA, you have the ribose sugar. Here we have the ribose sugar. Deoxyribose, okay, means here at the 2', okay, at the second position. At the second position, it lacks OH group. Instead of OH, you have only H. It lacks the OH group. Whereas in ribose sugar, at the second position, what is present? OH is present. Okay. So this is the difference. Okay. Now the DNA-dependent DNA polymerase, it cannot bind to the ribonucleotide because its active site cannot accommodate the 2' OH. Okay. So we already know that ribonucleotide, they consist of the ribose sugar, which has the OH attached at the second position, okay, of the ribose sugar. Consequently, it exclusively adds only deoxyribonucleotide and is specific to deoxyribonucleotide triphosphate, which is also known as DNTPs. Okay, so because of the presence of this 2' OH in the ribose sugar, in ribonucleotide, the DNA-dependent DNA polymerase only picks up the deoxyribose nucleic acids. Now, question number 10. Here we have to name few enzymes involved in DNA replication other than DNA polymerase and ligases. Okay, DNA polymerase, you know, it helps in the adding of nucleotide and also it has the exonuclease activity. Ligase, it helps to join the DNA fragment. Now, you have to name the key functions of each of the other enzymes. Okay, now the enzyme involved in DNA replication other than DNA polymerase and ligase are listed below with their function. So let's talk about helicase first. So helicase is the first enzyme that acts on the DNA. Okay. So helicase, it opens the helix, all right, which means it separates the nucleic acid strands, okay, for replication. So for the initiation of replication, or for replications to start, what is required? Helicase enzyme is required. So helicase enzyme come and it separates the two strands, all right, and then the replication begin, or we can say it opens the helix. So first enzyme is helicase. Then after helicase, we have the topoisomerase enzyme. So then topoisomerase enzyme comes, and what it does? It removes the supercoiling of the DNA. What does this mean? This means that it relaxes the supercoiling of the DNA. Okay, it relaxes the supercoiling. Okay, so first step, the DNA helix, it opens, and second step, what happens? The supercoiling of the DNA is relaxed so that the nucleotides can be added. Now third is the primase, or you can call it as DNA primase. Okay, so this is involved in the synthesis of the RNA primer. Okay, so this is also very important. First, an RNA primer is synthesized, and then the nucleotides are added at the 3' OH. Okay, and the direction of synthesis is always 5' to 3'. So this is also very important. RNA primer. This is of RNA primer for DNA replication. And fourth enzyme is the telomerase. This is also very important. What is the role of telomerase? It helps to synthesize the DNA at the telomeric end of the chromosomes. Okay, so this is very important. Otherwise, the ends, they don't get replicated. So telomerase enzymes, they specially synthesize the telomeric ends of the chromosome. This is also very important. Now coming to question number 11. Name any 3 viruses which have RNA as the genetic material. Okay, so if we talk about animal viruses, okay, mostly they have RNA, and that too single-stranded. Some plant viruses also, they also contain RNA, okay, such as tobacco mosaic virus. So you have to name these viruses. So before answering this question, let's understand what is the general structure of the RNA. So virus, okay, they are composed of nucleic acid, okay, which is usually RNA, and then we have the protein coat, which is known as the capsid. Capsid, they are made up of protein, so we call it as a protein coat. Okay, so the nucleic acid, okay, is surrounded by a protein coat known as the capsid. So I'll just drawing a rough figure here. Suppose this is the capsid here. So suppose this is a circular or spherical virus. So this outer is the protein coat. This is known as the capsid. And then we have the genetic material here. So this is the nucleic acid. We can say it can be RNA or DNA. Okay. It can be DNA also in some viruses. So here we have to name the viruses that has RNA as the genetic material. So in some viruses, RNA is the genetic material. The examples are tobacco mosaic virus. So this also has RNA as the genetic material. Other one is QB bacteriophage, or you can say Q beta bacteriophage. This also has single-stranded RNA. Again, human immunodeficiency virus, HIV virus. Okay, this also has 2 copies of single-stranded RNA. Okay, and it has 2 copies of single-stranded RNA. Okay, and the other one is influenza virus. So influenza virus is also RNA containing virus. So RNA containing virus, or the virus which has RNA as the genetic material, they are known as the retrovirus. Okay, bacteriophage. What is bacteriophage? Viruses that infect what does they infect? They infect the bacteria. Okay, one shape of bacteriophage is like this, tadpole shaped. Okay, so they have this hexagonal head. Okay, then they have this region, and then they have this kind of tentacles here. Okay, here is genetic material is present here, which can be RNA or DNA. So this is the nucleic acid. Okay, this one is known as the protein coat. So this is the capsid, which is the protein coat. Okay, so this is a type of a bacteriophage. Okay, so this is clear. All right, so this was the end of this session. We'll talk about rest of the questions in the next session. Thank you very much for listening.